Hello, my name is Dr. Jeremy Petrovich, Developmental Editor for Current Protocols at Wiley. It is my pleasure to introduce Dr. Ben Headley of the Victoria Hospital's Division of Special Hematology. Dr. Headley is a hematology scientist that holds a Master's of Engineering in Chemical Engineering and Chemical Technology from Imperial College, London, a PhD in Medical Biophysics from Western University, and is an American Society for Clinical Laboratory Science Certified Specialist in Cytometry. His previous administration and leadership roles include counselor for the International Clinical Cytometry Society, section lead for CLSI H62, validation of assays performed by flow cytometry, and is a reviewer for numerous scientific journals. Dr. Headley has published more than 25 peer-reviewed publications, and his work has been cited more than 1,100 times. With that, I present Dr. Headley for this webinar titled Initial Evaluation of the ClearLab 10C System in the Immunophenotyping of Leukemia and Lymphoma. Thank you.
Thank you, Jeremy. Today I'd like to talk to you about our evaluation of the ClearLab system that we did for Beckman Coulter. I have a few disclaimers that you can see here. The presentation is sponsored by Beckman Coulter. However, the opinions that I express in this are mine and not those of Beckman Coulter. Next slide. Typical reagent preparation involves taking individual antibodies that must be titrated and validated. These antibodies are re-evaluated when combined with other antibodies that may require further adjustment afterwards, as some antibodies do interact with others, causing undesirable reactions. The cocktail preparation is a time-consuming and quite a complex process, and individual antibodies are pipetted to create multicolor batches, which improves efficiency within the lab. However, errors in preparation can occur when pipetting large numbers of antibodies, and any errors that are incurred when making these multicolor batches incur increased expense and potential misinterpretation of patient samples. To this end, Beckman Coulter developed a ClearLab 10-color system. So it's a diagnostic screening panel for hematolymphoid cellular populations by immunophenotyping on a Navios or a Navios EX flow cytometer. This aids in the diagnosis of hematolymphoid malignancies, acute and chronic, lymphomas, myeloid, myelodysplastic, myeloma, and myeloproliferative neoplasms. And as for all laboratory-generated results, these findings should be interpreted along with other clinical and laboratory data. The ClearLab 10-color workflow is a sequence of steps that allows the laboratory to run the ClearLab 10-color product in a reliable, standardized way. This includes standardizing the way the instrument is set up, so with the use of FlowCheck and FlowSet beads. There's a compensation kit, which involves using beads, and the compensation is verified using an actual quality control process that Beckman Coulter has developed, which is one of the newer things that has been developed for this ClearLab 10-color system, which is a set of control cells that have standardized values and ranges so that there is a 10-color quality control product for this system. So in brief, the instrument is set up via this method here, which I've just outlined. The specimen is accessioned in the lab. The sample is stained on the tubes using this dry unitized format that Beckman Coulter has. The sample is lyzed and then acquired on the flow cytometer, and there are standardized templates for analysis available from Beckman Coulter with this 10-color workflow. So it is a complete system for the ClearLab 10-color. It is not just a product in this part here for sample staining. It is an entire process from instrument standardization through sample analysis. The compensation itself is done with a 10-color compensation kit using beads, which have a positive and a negative control bead within them. So the positive stains binds the antibodies and the negative bead does not. You can see here they're mostly CD4 antibodies with CD3 on ECD and CD8 on chrome orange. These allow for positive and negative beads to be used to calculate the exact compensation required for this 10-color product. The voltages and the actual assigned targets for these are given in the ClearLab 10 color system. So having a control material, which is one of the advantages of the 10 color system, is that there are control cells available with defined assay ranges. And you can see an example here of the Myeloid tube stained with the control cells. This is a Levy-Jennings plot, and it shows you here all the different parameters that are analyzed with the quality control package, and the limits here are the black lines, and the green is the mean that was given with the control cells. Depending on how the laboratory works, you would either develop your own mean and then run those, or you could just run it within the means. Even though this one, you can see here that one of these does run below the mean, it is still within the target limits. And this is what the control cells actually look like. This is the abnormal control cells. There's a normal and an abnormal. The abnormal contains about a 9 or 10% abnormal CD34 positive control cells. And then you can see here the staining for CD19, the staining for CD15, HLA-DR, CD13, CD38, CD33, and these are co-expression of CD117 and CD34, and co-expression of CD34 and CD123. So this allows you to run a quality control every day that you run a sample. If we then move to running normal blood on this with this system, you can see here's normal B cells run with the B tube, and this has obviously not solely B cell markers, but you're looking for abnormal B cell markers. But in this case, you can see CD10 only expressed on the granulocyte. You can see CD5 expressed on T cells, and here are the B cells that we're looking at here. If we look at bone marrow, you obviously see a different pattern. You see mature B cells that are polyclonal here. They express either kappa or lambda, but you see some pre-B cells here that are CD34 positive and the brightest CD10, and they do not express any immunoglobulin. They follow a normal maturation of brightest CD10, then dropping down their CD10 and gradually acquiring CD20. Once the cells lose their CD20, they start to produce their immunoglobulin, and by the time they actually are fully mature, they have lost all their CD10, and you see this polyclonal pattern here. If you look at normal blood in the T tube, you can see here you are staining for CD3 cells. There are some dim CD45 cells in here. These are actually basophils. You can see here there's the light blue are the T cells, so the CD4, CD8 tubes that co-express CD5. There are a few TNK cells. There are some CD3 negative, CD56 positive cells. These are the NK cells. They're not colored in this instance. This is just using the standard Beckman Coulter template, so it uses the Kaluza C software, which you can adapt and recolor populations, but it comes default with, I think, just 4 colors. So, but obviously there's a whole template within the system, so you can recolor populations should you wish. The templates are open so that they can be edited. They're not fixed. And you can see here the normal CD2 expression and the smearing of the CD7. The T2 does have gamma delta, TCR gamma delta in it, and you can see here some Treg cells here that express CD3 in that familiar pattern of the gamma delta stain. If you look at normal blood with the myeloid tube here, and this one's a little messy, but you can see the same dim CD45 cells here, 11B positive, mature granulocytes here, expressed in blue. The monocytes are expressed in green, and the lymphs are colored in red, sorry. And you can see here the normal familiar patterns that you'd see in normal blood with T cells, myeloid cells here, and the bright CD64, obviously the monocytes with 14 on them. If we look at normal bone marrow, you can see here some familiar patterns of expression with granulocyte maturation, as well as if you subtract out the monocytes here and leave the normal, the dim CD45 cells in here, you can see there are some precursor cells in here and some B cell precursors as well in this sample. normal blood with the second myeloid tube. Here we have CD123, which does allow you to identify these very bright CD123 cells, which are basophils. You can see the CD19 here, the B cells that are in the sample, and again the CD38, which is useful sometimes if you're looking at pre-B cells. If they lack CD38, then you can see if they're abnormal. And then you have your myeloid cells here with brighter CD33 versus dimmer for the granulocytes. And there's obviously the HLA-DR and CD15. So normal blood's quite easy to do. And then this is actually a panel from a paper that is in review right now that shows different levels, different samples run with the ClearLab system. So it's rather complicated, but briefly it's showing different diseases stained with the ClearLab system depending on what tubes. Obviously for what we did in the study, we ran all 4 tubes on all samples. However, if you have an inclination that is more lymphoid than myeloid, you'd probably preferentially run a lymphoid set of tubes on a sample rather than all 4 for cost purposes. But these 2 panels here, the A panels here show a CLL, it's 19.5, it expresses lambda. B panel here shows B-cell acute lymphoblastic leukemia, which admittedly with only the 2 panels is hard to tell, but it's actually a CD10 negative one that doesn't express any immunoglobulin. C here shows a TLGL that has bright CD2 and CD8 that doesn't express CD56. Here we have, in D, we have a T-cell leukemia that expresses uniform CD7, bright CD2, and does not express CD3, so surface CD3 negative. E shows an AML that's dim CD45, you can see it here, does not express has dim or some CD123 and 117 positive. F is actually a myelodysplastic bone marrow, type 2 with an increase in blasts. I think there were about 10% blasts in this one. G shows a monocytic leukemia that doesn't express CD34, but does express a little bit of CD13. Not shown here, I think they are 117 positive. And the very final one here is actually a myeloma patient that has a large increase in plasma cells that are bright 38. So, but that's actually not shown here. So next I'd like to go through a few cases that we had when we were going through the study that we used to evaluate this system. So case 1 is a 55-year-old male that presented with a lymphocytosis. You can see here he's got an absolute lymphocyte count of 14. He has an elevated white count. He has slightly higher hemoglobin than normal hematocrit. MCV is normal. Platelet count is low end of normal. You can see also he's got relatively normal neutrophil count. So peripheral blood sample was submitted for flow cytometry, and we used obviously our normal stuff, but we were evaluating the ClearLab 10 color panels as well, so we ran that at the same time. At the same time, we get another 50-year-old male with organomegaly, and we got a bone marrow submitted for him, but you can look at his blood counts here, and he has a similar picture. He has an elevated white count, he has an lymphocytosis, he has a little bit lower hemoglobin, but it's still normal. He's got a normal hematocrit. His platelet counts are a little bit low. So we have both of these samples, and we both used the ClearLab system to look at his the profile of both of these patients. So if we look at case 1, we have these similar sorts of pictures here. We have lymphocytes within the bone marrow or the blood in case 1, the bone marrow in case 2. They're smallish lymphocytes, but you can see a few little differences between the two. These are small, there's very little cytoplasm in both of them. A lot of these ones are clefted in this one here. So when we run the first case, we can see here the large amount of lymphocytes here. There's not actually a percentage on there here, but you can see here that these are a large expression of CD19. I think it works out to be almost all of the lymphocytes. they express CD20, they express CD5, a T-cell marker, and in this case they express CD200 as well, and they are mostly expressing immunoglobulin for lambda. So they do not express CD10, and they are dim to negative CD38. So the first case is a CLL based on the CD5 and CD200 marking, and then the second case is similar in such there's a smaller population, obviously because it's bone marrow, there's dim CD45, there's some of these B-cell precursors as well, but if we only look at the CD20, the CD10 here is not actually real. This is just a little bit of a bleeding of the lambda that is quite bright, pushing this 10 ECD up a little bit. You can see here that the CD5 positive, however in this case they are not CD200 positive. Express lambda, yes, and there's the 20, which is actually a little bit brighter than you'd see in the previous case. So this leads to a presumptive diagnosis of mantle cell lymphoma, which was confirmed by pathology after. There we go, just highlighting what we're seeing here. This is the difference here. You're seeing this lambda, which is actually quite bright, pushing up the CD10 a little bit here, which you have to be aware of in flow cytometry. Case number 3 is a 70-year-old male that presented to the ER with palpitations and bruising. Peripheral blood shows circulating blasts, so he has a slightly higher white count, still not at a critical level for our laboratory, it's under 30, but he has quite a low hemoglobin, a very low hematocrit, CV, very low platelets. He's mildly neutropenic. It says he's got a lymphocytosis, but he actually has these circulating blasts that you can see that were reported by morphology. Okay, so you can see here from the peripheral blood at low power and at high power, you can see these blasts that are they're quite large, but they have very fine chromatin and a very small amount of cytoplasm here. You can see the occasional small normal lymphocyte. And when you stain it with the ClearLab system, you can quite clearly see there's this large population of blasts that are within the blast region and this dim CD45 region here. They express CD19 and they express CD34. They're quite bright 38, which sort of identifies them as precursor B cells. However, these ones do not express CD10. They do not express CD5. They don't express either immunoglobulin. And if from the myeloid tube, from the second myeloid tube, you can see here that they abnormally express CD123, which gives a diagnosis of B-cell acute lymphoblastic leukemia because the cells were circulating in the blood. So case number 4 is a 32-year-old male presenting with circulating blasts on the peripheral smear. So a whole blood sample was then submitted to flow cytometry. Occasionally we do get the blood at the same time as the bone marrow in our laboratory, but sometimes depending on the time of day we get the peripheral blood first. And you can see here there's extremely elevated white count, very low hemoglobin, the patient is anemic, hematocrit extremely low, platelet count extremely low. The differential did report some normal lymphocytes, but the majority of them being at a ratio of 20 to 1 was the circulating blasts within the peripheral blood. And you can see here at low power, you can see an extremely hypercellular sample within the blood. And when you close in and see a little bit higher, you can see this is quite thick, but you can see here these blasts with almost no cytoplasm at all throughout. And these presumably, even though it's quite thick, these are the normal lymphocytes here, but there's a large number of these ones which are abnormal. So when you do the ClearLab tubes and actually look at the flow cytometry, if you run all 4 tubes, I'm only showing the one that's pertinent to this case. You can see here you have a dim CD45 region here that expresses CD5 but doesn't express CD3 on the surface. Expresses uniform and quite bright CD7 and co-expresses CD2. Does not express 16 or HLA-DR. Has some partial CD4 but no CD8, and does express CD3, and in accordance with fair number of these cases, there is abnormal expression of CD10 on this cell. So we have a T cell that expresses CD10 and CD34, doesn't express CD13 or CD64, which gives a diagnosis of T lymphoblastic leukemia in this case. Case number 5 is a 20-year-old male that presented with lymphadenopathy and a mediastinal mass. So this is I wouldn't say a typical presentation. However, it has been the case that the mediastinal mass is a bit of a giveaway with samples that come into our lab. The white count is not actually elevated in this case. The hemoglobin is decreased. The platelet count is dramatically decreased in this one. And the differential is quite unremarkable with a small left shift noted. But because of the mediastinal mass, as long as it is safe to, a part of the lymph node was biopsied and then submitted for flow cytometry. So this is a different sample type from any of them that you've seen before. When you look at the lymph node itself, you can see here a scattering of cells. When you go in, you can see these clusters of cells that are sort of sticking together, but they're all dark blue, very little cytoplasm within them. Again, all 4 tubes are run. However, you can see here, this is actually a lot smaller on the screen up here. On this one here, the CD45 expression sort of overlaps. It's not really that dim as it was with the case before. This one has a little bit reduced CD2 and CD5. It again does not express surface CD3. This one does not express CD34. It does have this uniform CD3 expression again, and this one does have the same thing again, no CD8 with variable CD4. The CD38 is actually quite bright in this case, and this one does again express CD10. But just to show, the red in here is the polyclonal kappa lambda. So these are some residual B cells in this sample from this mediastinal mass. You can see there are normal polyclonal B cells in this. And this is obviously with a little bit more workup and confirmation with pathology that it is a T lymphoblastic lymphoma within the lymph node. There was nothing in the peripheral blood. We did actually analyze that as well as part of it. Case number 6, this is a 60-year-old male that presented with severe anemia, which you can see here. Hemoglobin of 65, a hematocrit of 0.2, but there were reported blasts on the peripheral blood smear. So in this case, we didn't actually run the peripheral blood because a bone marrow was done after the results were seen. And so that was submitted for flow cytometry immunophenotyping. And we used the ClearLab 10C panels. In this, you can see in this case here, we have quite a hypercellular specimen. When we go in at high power, you can see these large cells that have some of this abnormal deposits within the cytoplasm here. And when we look at the flow cytometry results, we again see this very large population of dim CD45. The coloring is all standard within the templates, and for the most part, we never change the samples, but you can actually change the coloring. This purple population you can track throughout all of these different plots that are here. This one is not CD5 positive or CD19, so we don't believe that we're looking more towards a myeloid disease here rather than a lymphoid disease. And when you do the workup, there's CD34 positive, there's no surface CD3, there's no CD7, no 56, no 2, and this actually was an undifferentiated acute myeloid leukemia. You can see here's the rest of the workup showing the CD33, no 15, no real 64, 117 positive and DR positive, expressing 123, which is, I believe, common in about a quarter of all cases that we run. And they're showing the CD34, 117 co-expression here, and the associated normal granulocytes and lymphocytes colored in blue and red. The next case, which is the second last case, is another 70-year-old, this time a female that presented with pancytopenia. Obviously doing flow cytometry on the blood, even though there is obviously something going on, platelet count is relatively normal, but the patient is severely anemic, and there are nucleated red blood cells within the peripheral blood. It's more informative to get a bone marrow and have that one submitted for flow cytometry rather than to run the peripheral blood, which is our preference. You can see here, obviously, this is a very pancytopenic patient. There's very few cells in here, but when you can find some cells, even in the bone marrow, you can see here that they have very fine chromatin in them, these prominent nucleoli, and very, very little to do with any sort of granules within the sample. This one here is actually an NOS, acute myeloid leukemia, expressing 117, and here doesn't express CD34, does express 117, expresses CD64, again CD123, very weak expression of CD13, but quite strong CD33, and expresses CD11B. No T cell markers or B cell markers on this one. The final case is a 65-year-old male with known CLL disease, but presented with anemia and bone pain, which is not, I would say, normal for a CLL patient in my understanding. So a bone marrow aspirate was taken and then submitted for flow cytometry immunophenotyping. You see the white count here, which is more in pattern with what you'd think maybe with a CLL. You have the lymphocytosis here. The hemoglobin is decreased. The platelet count is in the normal range, though. So when we look at the sample here, you can see this is the bone marrow sample. You can see lots of lymphocytes within the sample, and you can see some other cells that are present in here as well. You've got these other cells that are present here, which look more like plasma cells. But the best way to do this is to look for the sample within the flow cytometry results and see what you get. So the patient is a known CLL, but you can find these cells. They express CD19, co-express CD5, they're dim CD20. They don't express CD3, obviously. They're 200 positive, and they express lambda immunoglobulin. So this is the CLL population that the patient has been previously diagnosed for. You can see when you look at the actual sample, you can see this consistent color. So the granulocytes are blue, the monocytes are green, the rest of the lymphocytes are red. And in this case, we colored the abnormal population black. And there's this dim CD45 region, which represents about 20% of the sample that is unaccounted for if you only look at the CLL population, which I'm sure no one would ever do in a flow cytometry lab. But you can occasionally see these multiple populations, multiple monotypic populations within a sample. And in this case, the patient is a known CLL. However, there's this rather bright CD38 expression here that doesn't express CD10 or 11B, may have some weak 117, although I'm not really sure of that, and doesn't express 5 or 3, and does have HLA-DR. And when you investigate this 20% population, you can see that it doesn't express CD19, but does express very bright CD38 and HLA-DR. Is not CD3 or CD5 positive, and does not express CD10 or 11B or 117 or CD15, and is another monotypic population. So this patient was diagnosed with myeloma as well. So when we look at the entire set of samples that were run for this evaluation across 4 different sites, I've given you 8 examples of some of the sorts of samples that we saw from blood and bone marrow and lymph node tissue. We actually did run across all 4 sites a total of just over 450 samples. Obviously, you want to run some with no malignancy and some with malignancy. This is a spread of what we saw. Most of them were non-Hodgkin's lymphomas, then chronic leukemias, a fair number of acute leukemias, so that's 1/9. So that would be around about 10% or so, 11% of the samples were acute leukemias, a fair number of plasma cell neoplasms. MDS, MPN, and otherwise categorized samples. And you can see it was pretty consistent across all 4 sites, trying to get a good spread of things that we're seeing. You can see in the pie chart in the top left here the actual spread percentage-wise of all the different types, which fits with what you'd see in most larger flow cytometry labs, where you see the predominance of non-Hodgkin's lymphomas, then chronics, then acutes. And of those acutes, most of those, depending on if you see lots of adults or pediatric samples, you could have a shift towards myeloid or B-cell types of acute leukemias. So when we look at the actual table to see how well the ClearLab panel did, so if the clinical diagnostic outcome was determined from a range of tests done within the reference laboratory, so lots of these patients were diagnosed with disease with multiple modalities, so pathology, cytogenetics, all of these clinical findings all coming together to give a clinical diagnosis. And when you strictly take that, which is a whole panel of things, and compare that to just the ClearLab by itself, in the absence of any clinical data, you get pretty good results that show that the ClearLab itself can detect a malignant versus non-malignant sample, showing that the presence or absence of a malignant population can be found with the ClearLab 10 color panels just solely by itself, which is very good performance. Obviously, in a routine laboratory, you take into multiple factors, so you'd have the aid of lots more diagnostic things. So even by itself, the ClearLab can detect populations, abnormal populations, or in this case, eliminate the need for further testing. So here we have the 10 non-malignant and the 18 absence of normal and abnormal phenotype that the ClearLab detected. So in conclusion, the derived reagents that we used for this study were very stable. The product itself took 9 months to complete, and I believe that the reagents now have an 18-month stability guide once they're received. This is not true once they're opened. They're in resealable pouches, and they are guaranteed for a month, I believe, after they are opened. The system itself was very reliable throughout the whole process that we did throughout these 9 months. The system itself is completely standardized from instrument setup through quality control, daily quality control, the reagents, and to the analysis, all the way to the reporting. There is standardized templates for everything, including the quality control, which is probably one of the more innovative products that we've used in a while. This is a true process control. The reagents themselves are obviously kept in the fridge. They are stable for, I believe, about a month or 6 weeks, and they are set up and stained exactly the same way that you would set up and stain and lyse your actual samples. And then they have, obviously, like I showed, the Levy-Jennings report, so that you can track over time exactly how your instrument and your quality and all your reagents are working. There is an educational component to this as well, because if you look a little further down, there's an educational casebook that goes with this that was born out of the study that was done with the 4 flow cytometry labs. And this is an excellent training guide for staff and students. If you have any new staff, it's a good way to learn about flow cytometry, depending on how much you've seen before. It's also good for residents, for them, because there are morphological pictures in it as well, I believe. So it gives them a complete picture of what a flow cytometry lab does. So the results from the clinical study did show very good concordance between the labs and the actual results from the multidisciplinary approach done at each site. It was quite a tough bar to set that the ClearLab itself would show good concurrence with a large multidisciplinary approach that involves pathology, clinical, and genetic data, to say that a single set of 4 tubes from flow cytometry would be able to push you in towards the right direction to say that it was the same as all these other things. But in the end, you would never use this product as the way that it was set up for the study, because a definitive diagnosis really does require this multidisciplinary approach where you merge the flow cytometry data with morphological data, clinical data, and more and more frequently the genetic data that you get from the samples that you get received in the lab. And finally, I'd like to thank all the people in the lab that have made this all possible. My former mentor, Mike Keeney, who allowed me to learn all of these things. My two medical leads, Dr. Chinyi and Dr. Shah. My other retired, recently retired mentor, Wendy Brown, who taught me much morphology and much general hematology, and all the members that are past and present in the flow cytometry lab, some of whom have moved on to other things, others who do research, and others who have also retired. And this is a picture of our lovely site here on a nice spring day last year. Thank you very much.
Thank you very much for that webinar presentation. So our first question from the audience involves the rationale of the antibodies included in their combination for the 4 10-color tubes. So can you just elaborate on how these markers were included, what kind of tubes were initially evaluated as well, and what the fluorochrome selections involved?
Sure, Jeremy. The antibody combinations were developed actually quite a long time ago, and they were based on the Bethesda guidelines, the WHO originally 2008 actually, but then revised with the 2016 guidelines and some other publications as well. They were done with in consultation with a fair number of people actually. I don't know all of their names, but I know that there were a lot of people involved in it, and this actually we actually have a publication that is in press right now, so that will detail maybe a little bit more about how the combinations were come to. But the brief answer is yes, the Bethesda guidelines from the WHO 2016.
Okay, thank you. So for this assay that you've been discussing, is this something that can also be run on other instruments, or is this specific to the Navios flow cytometer?
Yeah, so because this is a Beckman Coulter project, sorry, not project. It was a project. The assay has only been run on a Beckman Coulter system, a 10-color system, either Navios or the Navios EX. There's no reason why it probably couldn't if there were the right filter configurations, but the advantage of this assay really is that it has been FDA cleared and is IVD. So if you were to run it on a different system, if that is definitely feasible, you would break the IVD status on that. So usable, yes, given the same configuration, whether you would want to, that is a laboratory choice or not. So, but no, it hasn't actually been tested on something else.
Thank you. One of the audience members is asking, why is CD23 not included in the B-cell tube?
Well, that was a decision made more recently than some of the other markers. There's a lot of discussion in the literature whether CD23 or CD200 should be used. Depending on which publication you read, you can get better results with CD200 or you get CD23. Some guidelines say that 23 is better than 200, others say the inverse. They both do the same job. They differentiate CLL from mantle cell. I think 200 myself is better. We use 200 in our laboratory. but I know that there are other labs that use 23 and believe that 23 is better. I think that we have seen a little more consistency maybe with the CD200, but that was a decision made by Beckman Coulter, and I think it was a good one.
Thank you. So how do you integrate this with other LDT panels? Specifically, do you use the same instrument settings or not?
Yeah, so the way that this is set up, the Beckman Coulter do supply all the setup and everything really required to run these panels, and so they're IVD released, so that you really have no the way that's set up is the way the manufacturer intended, and it has I would have thought it would have a guaranteed way that it works, which is the way that we've used it. And it's a 10-color panel, so it uses all the detectors on the instruments that we have. There's no reason why you couldn't run other 10-color panels with those same settings. There are some caveats to that, like you would have to be running the same fluorochrome. So these run PE Sci 5.5 and APC tandems off of the red laser. Obviously, the PE Sci 5.5 is off of the blue laser. But if you were to run different fluorochromes, and the specific blue off of the violet laser, if you run different fluorochromes, they do emit differently. So it will affect your compensation. So all things equal, you could run them with the same settings. Yes, would it require a little bit of manual adjustment? More than likely, if you have used a very different fluorochrome. If you use something that is substantially brighter, then it could affect the compensation that you have. So yes, you could, because I understand that yes, running multiple settings files is arduous and having to set those up. But we do run different numbers of antibody combinations on 10-color settings files, so we don't necessarily run 10-color panels all the time with on a 10-color settings file. So we run 6, 7, 8, 5, numerous numbers on 10-color settings files. So you'd have to evaluate how your LDTs would work on those instrument settings. But it's definitely worth exploring, because it does mean that you don't have to set up your instrument for each individual panel, like your LDT, as well as this ClearLab 10-color panel. Hopefully that answers your question completely.
Great, thank you. What is the limited detection for the ClearLab 10C panels? And can you elaborate on how that limit is calculated?
Yeah, so the everything for this product was calculated based on the study that I sort of showed the brief version of at the end there. So the settings, the way that they're set up, is that it collects 50,000 events, and the limited detection was back calculated then to be 1%. You could actually go a little bit lower, but 1% is what was submitted to the FDA, so that's the limited detection. You more cells that you count, the limited detection would go down, but this is not intended to be more than a diagnostic panel, so it's not intended to do residual disease type assessments. That's why it was set up the way that it was set up. So it wasn't arbitrarily established, it was actually determined experimentally from the number of events that you can possibly collect within a small space that would be clonal and indicative of disease. So wasn't just arbitrarily decided, no, it was calculated. So.
Thank you. If you do AML MRD, would you use the M1 and M2 tubes in the panel with 1 or 2 additional tubes, or do you have to develop a separate AML MRD with total disregard of the M1 and M2 tubes?
Yeah, that's a good question. I've been asked that one before. AML MRD is not simple. depending on which way you do it, whether you do a leukemia-associated phenotype or whether you do a difference from null. The M2 tube could be used. I've been asked this before. It could be used to determine whether you have AML MRD. That is not the intent of these tubes. That is the actual. I believe the legal disclaimer probably written on the tubes would say that, or on the package would say that this is for diagnostic purposes and not for doing MRD assessment. It's not to say that you couldn't use them, but it would then be an LDT. It wouldn't be an IBD test. The M2 tube does allow you to do that sort of analysis. I mean, it has the markers in there. It has, DR 34 and 117, and it has 38. It allows you to distinguish, between, maturation of blasts. You could do it, but like I said, it's not what the tubes were designed to do. If you have your own AML MRD panel, then that would obviously, there's lots of them published everywhere. There are, I mean, it's possible, yes. Whether you could do it with just a single tube, that's debatable. Lots of people do it now. So obviously, the M1 and the M2 tube do give you a complete picture of what is happening with the myeloid tube. So yes, you could do it with both. And given that the CD34 does cross both, and the DR and the 13, you could probably quite easily match between the tubes and hopefully locate your, or not locate your residual disease population. So yes, it is possible, but no, it is not part of the actual product that really is being discussed here. But definitely an off-label use potentially. So.
Great, thank you. Can the ClearLab control cells be used on other flow cytometers?
They've only been validated on a Navios and a Navios EX, right? Like that's the standard answer. There's no reason why they probably couldn't. They're process control, so they work as you would work with as samples do. So they are stained or washed. If you use the B tube, they're washed, stained, lysed, washed again, and then put on the machine. I don't know how they would behave with other fluorochromes, with other products, because they have been developed specifically for these panels. So, I mean, as a theoretical, yes, they could be used on another flow cytometer. Have they ever been? Not to my knowledge. But the percentages, the package insert, all of those things that come with the control cells are all developed for the ClearLab 10 color, not for any other product. It's not a product that you can just say you can use just for running your own LDT. I mean, there's no reason why you probably couldn't evaluate it, but again, it would be breaking the IVD, or it wouldn't be IVD approved. So,
thank you. So someone else in the audience is curious as to why CD34 is present in all four of the tubes.
I think I sort of alluded to that one before. The fact is that the CD34 allows you to go between the tubes, especially if you have a blast population that expresses CD34, or if you're looking at a bone marrow that has CD4, you can stitch all the tubes together, and it allows you to sort of see how your tubes are performing all against each other. So we do a sort of a similar thing with some of our tubes as well. So it's a very useful way to anchor your tubes, well, your patient between tubes. So that's why it's in there.
Thank you. How are the analysis templates provided? And somewhat related to that is, what analysis software did you use in this study?
The analysis templates were provided by Beckman Coulter, and I believe they're downloadable when you have the product. They use Kaluza, which is obviously Beckman Coulter's proprietary software. They use a different version now called Kaluza C, which is very similar to the research Kaluza that has been around for a number of years that many people use. And the templates have almost single thing that you could imagine as far as, gating is required. Whether you need all of them to report, that's a personal preference, but they're provided, so you can obviously change them at your will. I don't know quite Beckman Coulter would be able to answer the questions on whether you could change the template and whether that would affect your IVD status. I'm not sure. So, but yes, they're provided by Beckman Coulter. They're very easy to use. And they work for the control cells as well. So.
Great. Can you speak to the cost effectiveness of this product, especially versus homebrew? I think just user-generated panels, I suppose.
Yeah, I mean, that's always the question, right? I mean, typically, if you can make it yourself, it's cheaper. I mean, I can't speak to that because I am not a Beckman Coulter employee. However, what you're getting with this is you're getting the whole system and you're getting an IVD approval. So for very large labs that have already gone through, many iterations of what they're doing and deal with, hundreds of samples a day, may not be something that they particularly would want, but smaller labs that don't have the knowledge or the expertise or, the history behind this sort of thing, it does allow you a complete system. So set up all the way through control, which a lot of 10-color people doing 10-color LDTs, you do need to have some sort of control material and through analysis. So it does allow that part. So I presume you would pay a premium for that. It's one of those things. You pay for the R&D done for it. So, but yeah, talk to Beckman Coulter. I'm not the right person.
So in one of the cases, there was a very sparse population of plasma cells that were CD38 positive. What is your view on the HLA-DR expression in this case? Is it a true positive or possibly appears positive due to spillover of bright CD38 in the HLA-DR channel?
Oh, I would have to answer that one offline to the person. It is entirely possible. You do have to be a little careful with very bright antigens in any panel. So I don't have the PowerPoint up here. But yeah, I will answer that question offline to the person. But yes, it is something you do need to be aware of when you have extremely bright fluorochromes and whether they are spilling over into the next channel. You have to be very aware of that one if you have immunoglobulins after something that could be extremely bright. So yeah, but I will have to look at that one. Sorry, I don't have the presentation up right in front of me right now.
When you have an acute leukemia case, when do you decide to add an intracellular tube?
We typically do it once, if it's a diagnostic, and well, if we know it's acute leukemia, then we have to lineage type it. We don't assume anything. Seen enough weird stuff in the last 2 months that, that confirms that. So yes, we don't always do them at relapse because we already know what we're dealing with. But yes, we do them in some of the cases that you saw that I presented. Yes, we would have to do an intracellular tube, which is not available currently with the ClearLab 10 color. So yes, there is still a requirement to do an intracellular tube. We do it every time we get a new one.
All right, great. Thank you very much. We've reached the end of our time today for questions. I'd like to thank Dr. Ben Headley of the Victoria Hospital's Division of Special Hematology for taking the time to present to us this webinar regarding his usage of the ClearLab 10C system. Any questions that were not addressed will be answered by email when able. This presentation was sponsored by Beckman Coulter in partnership with Wiley's Current Protocols and will be available for on-demand viewing on the Current Protocols website at www.currentprotocols.com in the webinar section under resources. Again, thank you, Dr. Headley, for taking the time today and for the great presentation. Thank you, everyone, for attending.